This is Environmental Lab 6802. As you enter, as you can see, this is our particle size analyzer. This is a sink. Some of the stops are here, will be thrown out, and then you get the few mode, and there are a few stops here, but pretty clean. And then you have this high temperature oven, which will heat up the stops more than 700 degrees Celsius. And then you have some old columns, and if you look right, this is our sample process. Inside you have this Sony cutter, and so this bench is just for sample processing. You have this vacuum setup for any extractions, and then over here all the DNA stuff. On the bottom you don't have much stops, you can sit there, and if you go across, this is for microplastic setup, where we have all the experiments going on related to microplastics. And underneath you have some sand and soils or biochars, iron filings that you need to use. And the pumps are here inside it, and then this is our IC, ionochromatograph, and then we have, this is a storage for sieve and stirrer plate, some of the cubes, and we have this sink, another sink, which is pretty dirty at the bottom. We can clean that later, and there are some stuff here. And this is walk-in refrigerator. This walkway is free, as now we don't have any issues. We have some old columns that can be used later, and then you have this setup is for compactions and other experiments related to Caltrans. And if you look around, this is our EV analysis, and so this area should be clean so that people can sample analyzed. And this bottom has to be enough space so that people can sit here and do the processing. And you have lots of plastic, lots of glassware, bottles, and this is for bacterial analysis. So this is this has the flame and everything else. So pH meter. We have lots of box of soil moisture retention curve analysis. So those boxes will be open, and we'll put the instrument here. And the one last thing to do here is we have these some unsorted items from different students. So those will be sorted out. So this area should be free, not too clean. So we leave it there. And the very last one, we have the samples from Benji. So we'll move it out eventually. So that's it. So as you can see, the slab is pretty clean. So this would be our standard to maintain it. So I'm pretty happy that we all help out to clean the lab. So now this is ready to really work more efficiently. Thank you all for helping. Bye.